Thank you very much. As a retina specialist with 29 years of experience in leadership roles in academia and in industry, I'm excited to present an introduction of Ikarovec to you all. At Ikarovec, we're building a platform of bicistronic gene therapy. So what this means is that one vector produces two therapeutic proteins which have some degree of synergy. And our first therapy is a bifunctional gene therapy for geographic atrophy. What differentiates this program is that it not only will preserve retinal anatomy, but we believe it will also preserve visual function. This is the core team, and I just want to highlight that Katie Binley is our Chief Scientific Officer. She has 25 years of experience in gene therapy, previously working at Oxford Biomedica. The precursor company, Quithera, was also working on bicistronic gene therapy, headquartered in the UK, and it was acquired by Astellas a few years ago. All of you know Dave Geyer. He's our board chair. Jean-Philippe Cambal is also one of the board members. He's a well-known executive in gene therapy in the UK. He's currently CEO of Avet. He is chairman at Splice Bio, which just had a big Series B, and he's been COO at GenSight. All of you know the limitations of current therapy. What sets our therapy apart is that with our multi-pathway approach, we can preserve not only retinal cells, but retinal function. And of course, with the gene therapy, we address high treatment burden. And finally, you'll see in a moment that one of the two therapeutic proteins is a potent anti-angiogenic agent, which can prevent neovascular conversion. So this is a basic schematic of the gene construct. On the right, the gene therapy produces complement regulatory protein CD46, which has been shown to be reduced in eyes with AMD. By making this protein, we can reduce complement activation and also reduce our retinal cell loss. On the left, the construct also produces PEDF. This has also been shown to be reduced in eyes with AMD, and it has a myriad of functions, including being retinoprotective, anti-inflammatory, anti-angiogenic, and reduces A2E accumulation. Normally in the eye, the RPE cells produce it, and it works in an autocrine and paracrine fashion where it supports phagocytosis of shed rod outer segments, thereby reducing drusen accumulation. I should add that PED has these interesting aspects, and when I first started looking at the data, I was asking why nobody's using this as a recombinant protein and injecting it periodically in the clinic, and the answer is that PEDF has a very, very low or short half-life of about 35 minutes, so it almost has to be produced as a gene therapy. So in summary, for this first asset, we're not only working on complement activation, but also on inflammation, oxidative stress, lipid deposits, and drusen formation. This is a busy slide, but it basically summarizes 5 years of platform development by the team in the UK. I divide the models up into three. There's complement modulation models, there's retinoprotective models, and there's angiogenesis models. And across these models, we see a consistent benefit of our vector. Interestingly, we also see preservation of retinal function. And then when we compare our bicistronic vector to monocistronic vectors containing either of the two components, we see added benefit. And when we go head-to-head with competitor constructs that produce CR1, CD59, or CFI, we see superiority in these animal models. We have non-human primate data showing that this vector is safe and well tolerated, that we have therapeutic protein levels being elaborated in the vitreous. You can see the immunohistochemistry on the left with robust expression of PEDF in the RPE and in the retina. And on the right, via RNA-scope, we see transgene and vector expression. And on the far right, you can see it's underlying the fovea in this non-human primate macula. Our core clinical trial design will involve a 3x3 dose escalation study followed by dose expansion in the two highest tolerated cohorts. We plan to enrich for fast progressors and also have readouts as early as 12 weeks where we'll see clinical safety, we'll see therapeutic protein expression via aqueous taps, and we'll also have a host of biomarker readouts, both functional and anatomic. I mentioned we're developing a platform, so we're also looking at capsid optimization for office-based delivery, and we're also developing a second asset for wet AMD. This just gives you a flavor of some of our capsid optimization work Here you can see an optimized capsid for intravitreal delivery. On the left, you see this retinal hole mount with widespread expression of M-cherry. In the middle, you can see M-cherry being expressed not only in the ganglion cell layer, but also in the inner nuclear layer. I mentioned that we're producing a second asset for wet AMD. Here, this vector produces not only an aflibercept-like molecule, but importantly, an anti-connective tissue growth factor single-chain variable fragment. And this targets a key driver of fibrosis in wet AMD. We all know that anti-VEGFs don't prevent fibrosis, and fibrosis is what actually drives long-term outcomes. I was an investigator in the CAT trial, and the CAT trial showed that half of the patients on therapy developed fibrosis by 24 months. So in summary, we're developing a bifunctional platform with ICAR1 addressing geographic atrophy via neuroprotection and complement modulation. We think that can address visual function in geographic atrophy, and ICAR002 for wet AMD with an anti-angiogenic and a potent anti-fibrotic. We're also working on office-based delivery, and we're about to embark on fundraising to support our GMP manufacturing as well as clinical trial. So thank you very much for your attention.